So finishing up chapter 20, the next thing we're going to talk about is the resistance to antimicrobial drugs. And so we talked about in the last chapter how beta-lactamase drugs are beta-lactam drugs are blocked by beta-lactamase, which is an enzyme that some bacteria carry with them to ultimately block the penicillin, ampicillin, amoxicillin, all these type of drugs. Now, one of the other things we talked about is that this is actually happening because of horizontal gene transfer. And so some of these horizontal gene transfers are coming from the misuse of antibiotics. And so some of the stats that we have is before penicillin G came out, or when penicillin G first came out in the 1940s and 50s, only about 3% of all Staph aureus was resistant, and now more than 90% of it's resistant. And one of the biggest problems is that hundreds of tons of penicillin are used each year. Many countries do not require a slip from the doctor to a prescription to actually receive these antibiotics, and so a lot of people are misusing them quite a bit. So let's say you have an infection and that you're supposed to take the Z-Pak for a maximum of, or for a minimum of 5 days, but you decide after 3 days you're feeling a little bit better, and a lot of us are going to forget to take those last 2 pills. What's going to occur is what you see here on the right, and in few people, not everybody, of course. And so you can see here that the S stands for sensitive, meaning that the drug is going to take them out, and R stands for resistant. Well, you're going to have a couple resistant ones at any given time, but luckily the antibiotic is going to kill off the sensitive ones, leaving your immune system to basically clear out those 1 or 2 resistant ones that are resistant to the drug. It doesn't mean the immune system can't take them out. The problem is that if you don't stay on those antibiotics, you're going to start seeing that resistant one starting to increase. The sensitive ones are going to come back, and the immune system is going to quickly become overwhelmed. Again, remind yourself that these bacteria are replicating every 20 to 30 minutes. And so it's not going to take very long for them to grow out of control. Now you have a much bigger problem because all the bacteria are going to be, or you're going to have a much larger portion of bacteria that are resistant, mainly just because there was misuse of the antibiotics. So some of the mechanisms of acquired resistance. This means horizontal gene transfer, or it could be through mutation. One is going to be drug inactivating enzymes, so that's going to be the beta-lactamases that basically block the ability of the drug to do any work. Other ones are going to be altering the target molecule, and so these are going to be ones that are going to change the structure of whatever the target is that that drug is looking for, and by changing it, it's going to prevent the drug from being able to do its job. Others are going to decrease the uptake. So if it recognizes that it's pumping in a drug and it recognizes that drug is not useful, it will shut down those pumps and prevent that drug from actually getting in. And then also they can go ahead and increase the pumping out, where in this case, if it recognizes that it's toxic, it will actually go ahead and pump the drug completely out of the system as fast as possible. So acquisition, spontaneous mutations are only going to occur during replication, and that's because bacteria, well, let's back up. DNA replication is going to happen whether you're in a prokaryote or eukaryote. It's just part of the cell cycle. And accordingly, as the DNA polymerase is going through replication of the DNA, it's going to make mistakes about 1 out of every 100 million or so nucleotides. And so a lot of these mutations aren't going to yield any problems. But if that's occurring and you have billions of bacteria in you, the likelihood that a couple of them may actually have a strange spontaneous mutation that yields resistance to a drug is there. The percentage is pretty good. And so we want to try to stop this from happening, especially if you're just on a simple drug that you're only taking one and you are not being very efficient with it. Well, you're putting small pressure on that bacteria. You're not putting a lot because you're not taking the drug properly, but just that little bit of pressure that you're putting on it will actually force mutations to happen at a faster rate. thus increasing the chances of getting a resistant population. Therefore, that's why we talk about combination therapy being more important. Multiple drugs at once is going to make it very unlikely for the cell to develop resistance to both drugs or whatever number of drugs you're using at the same time. Gene transfer we talked about in the last chapter. The gene transfer is going to be transferring the genes through the sex pilus. This is what we call conjugation, mainly transferring plasmids between different bacteria just through the process of forming a sex pilus, which acts like a bridge between the two bacteria, and the plasmids are able to transfer back and forth. Now, remember that most plasmids are carried in multiples inside the bacteria. Upwards to hundreds of copies of the same exact plasmid can be carried in a single bacteria. And so it's not a big deal for it to give up one or two of them to a neighboring bacteria to allow that bacteria to get the resistance too. So we call these the R plasmids, and the R just stands for resistance. So examples of emerging resistance, the E. coli, which is part of our normal intestinal tract, is starting to become resistant to a lot of our beta-lactam drugs, and this becomes a problem only in people that are immunocompromised. Typically, a healthy individual is going to have their E. coli kept under control, but somebody that might be immunocompromised in some sort of fashion, their E. coli can start growing abnormally in the intestinal area and start causing serious problems. And so the fact that we're having some resistance in this is disconcerting, especially since we've already seen vancomycin resistant E. coli, which is not good at all. Staph aureus, of course, is the most famous. We got methicillin resistant and vancomycin resistant. Typically, it comes down to where it was acquired. So if you acquire the MRSA in a healthcare association or a healthcare situation, it tends to be antibiotic resistant, especially against methicillin and penicillin. And so usually we have to get vancomycin for people that acquire MRSA in the hospital. Unfortunately, we're also seeing vancomycin resistance in these areas too. So we have very strict guidelines to try to halt the spread of these, mainly quarantine and whatnot, whenever somebody pops up with a vancomycin resistant Staph aureus. Now, the reason why it's so much uglier in a healthcare is that you basically are passing this bacteria around through basically you're passing the bacteria around through immunocompromised people. Again, people don't go to the hospital that are typically healthy unless they're just coming to visit or coming to work. The majority of the patients there are there because their situation is bad enough that they're in a hospital. And so this is not a good thing, but this is mainly why we see it as healthcare association and not community acquired. Community acquired MRSA is just spontaneous pop-ups of MRSA, and usually we can treat it pretty effectively with other third-gen drugs, including vancomycin if we have to. Now, tuberculosis is one that makes me nervous because you have to be on this therapy, combination therapy, for a long period of time, sometimes 6 months or more, and a lot of patients aren't going to comply. Compound this with the fact that some figures say that 1/3 of the world's population is infected with tuberculosis. Granted, it's latent, which means that the person doesn't know they're infected. But because 1/3 of the world's population does have tuberculosis or is suspected to have tuberculosis, and it takes 6 months to treat, and many patients aren't going to comply, we are starting to see some serious multi-drug resistant tuberculosis popping up. And ultimately, we're getting extensively drug resistant tuberculosis, which basically none of the drugs that are available work whatsoever against it. And so this is becoming very scary for a global threat situation. So what do we do to slow this all down? Well, us researchers are going to continue to come up with novel drugs, continue to discover new things nature's already provided to us, and hopefully be able to always stay at least a half step ahead of the microbes. What it's going to come from for people in healthcare and from physicians is to force patients to understand that they must take these drugs properly, that they can't take them out of sequence. Whatever the prescription is, it has to be followed. And to doctors, make sure that they're only prescribing antimicrobials to people that we are sure have bacterial infections and not just a virus infection. And not because we're just trying to push that patient out of the doctor's office by fulfilling whatever desire they have for having a medicine leaving the doctor's office. We're just going to have to tell them, sorry, it's a virus and they need to get over it. Patient is going to have to follow the instructions, even if it's inconvenient, especially something like tuberculosis that you have to take the medication for several months. It's important that you take it at the same time every day, because we're trying to maintain those blood levels to the levels that we prescribe to you, and so, or to the patient, so they have to follow it to a T to try to prevent the resistance. So we have to educate the public, not just patients, but the public, and let people know that antibiotics just do not work against viruses. They cannot cure the common cold, so you do not need an antibiotic unless it has switched from being a virus to a secondary infection that potentially could be a bacteria. Then, of course, you're going to need an antibiotic. But mainly, just because you started feeling bad yesterday and you have the sniffles, you do not need to take an antibiotic. It's just completely a waste of your time. Global impacts on the use of antimicrobials. We're starting to see resistance, mainly because bacteria have no political boundaries, and all it's going to take is one person traveling to bring a microbe here. So that's what makes it so nerve-wracking about extensively resistant tuberculosis. The areas that we find that in is India, Pakistan, and Afghanistan areas of the world. And so it doesn't take very much for one of those people to get on a plane that may not be doing it intentionally. They just are coming here for business, for family, for just pleasure, and they have no idea that they're carrying extensive resistant tuberculosis until it's too late and he or she has already infected several other people. So we have to kind of pay attention to this and be aware that we can't shut down borders. We can't stop people from getting on a plane. It's just not feasible. Other issues are going to be the fact that I already pointed out that many of these antimicrobial drugs are available without a prescription and lots of places in the world, and that may allow for improper use, and so we need to try to find ways to at least educate the public if they're not going to change their ways. We also need to get antimicrobials out of our animal feed. Unfortunately, it's kept in the animal feed at low levels to try to prevent the animals from getting sick, but instead keeping them at such a low level actually selects for the drug-resistant microbes, because again, we're putting pressure on them to change to try to get around these antibiotics. So a lot of resistant salmonella strains have been linked to pigs and chickens that have been fed antimicrobial drugs during their entire lifetime. Now, switching over to viruses are incredibly hard to target because they're mainly found inside our cells, and so to actually target a virus means that we have to get inside our cells and try to attack it, and this just isn't very feasible. So there's not a lot of antiviral drugs out there, but yet we still have several that are on their way or have been able to be successful, and those are the ones we're going to focus on. So just as a reminder from chapter 13, the virus, how a virus works is that they have 5 major steps. They have to have entry, then they have to uncoat, then they have to synthesize their nucleic acids, then they have to reproduce new virus particles, and then they have to exit. And so different drugs are going to focus on these 5 different steps. So some of the new HIV drugs that are out there are trying to block the entry of the virus into the new cell, trying to protect those CD4 cells. So the 2 drugs that you see here are part of the cocktail that's going to basically try to prevent the HIV from binding. I told you that one of the receptors is CD4, so the CD4 T cells seem to take the brunt of the punishment from the HIV. However, there's also a co-receptor, CCR5, that HIV can also bind to, and so these 2 drugs essentially block their 2 big receptors. For viral uncoating, these drugs are going to be there to block the virus from being able to uncoat. And so the viruses that actually have a protein coat, these two drugs are pretty good at blocking their ability from getting the nucleic acid out of the coat. And so here you have one that blocks the flu from being able to uncoat, and that's going to be useful for like Tamiflu, essentially is what this drug is. Unfortunately, because it is viral uncoating, that means that you've already been infected. So most of these drugs that are found here are going to be very limited to just basically minimizing the number of days that you're sick. So I think Tamiflu moves you from being sick for 5 days to like 4 or 3.5. It's really not that big of a deal, but it's a start. It's something that we're starting to truly understand how viruses work and ways to actually prevent them or to cure the patients that may be infected with a virus. Now, nucleic acid synthesis is pretty difficult to deal with, mainly because they use our machinery to essentially replicate their nucleic acid. However, there are a couple virus enzymes that are very useful to target. The one that's the most popular is acyclovir. This is one that's used to treat herpes and leaves most of the unaffected cells safe. So it does have some very significant side effects. So you're not going to give acyclovir to somebody with a cold sore. You're going to give acyclovir to people that have genital herpes, and usually somebody that actually has frequent outbreaks, not somebody that gets an outbreak once a year. This is usually a drug that's going to be minimized to people that get monthly outbreaks, just to try to give them better quality of life despite the side effects. Some other ones here are going to be useful for specific viruses. So the first one is against cytomegalovirus infections in immunocompromised patients. This is going to protect them usually from losing their eyesight, and worse, and even more so their life. Ribavirin is actually used to treat infections in newborns. So this is a drug that we give to people, to newborn patients that are infected with RSV. One of the other ones that we can look at in HIV is that HIV is a retrovirus, which means that they come in as RNA and they have a special onboard enzyme that allows it to go from RNA backwards to DNA. And so these transcriptase inhibitors are useful in treating HIV, mainly because we don't have reverse transcriptase in our body. And so this is a good target for trying to minimize any sort of side effect issues. Integrase inhibitors, this is going to be good for preventing the virus from inserting DNA copy of the genome into the host cell. A lot of viruses like to take free rides by going ahead and incorporating their nucleic acid sequences into ours, and so every time our cell goes through replication, they get replicated for free essentially. And so that requires an enzyme called integrase, and so these inhibitors are going to hopefully block that ability of the virus. For the fourth step, having to assemble and release the viral particles, we have a couple different opportunities here to attack. One is a protease inhibitor. This is going to block any proteins that were translated during the process of replication of the virus, and so there's a large sequence of different drugs there that are good at that. Neuraminidase is specific to flu. Remember, we talk about H1N1, H being hemagglutinin, N being neuraminidase. Well, these are surface markers on the virus, and these neuraminidase specifically is needed for release from the host cell. Well, if I block neuraminidase, the virus can't break free. It's trapped inside the host cell, and it's not able to move on and infect other cells in the body. Now, eukaryotic pathogens, luckily, as I said at the end of chapter 16, we don't worry about eukaryotes as much, mainly because they are not very pathogenic in healthy people. However, we do have a couple issues here that we can focus on. But again, the toxicity tends to be pretty high in these, so these are only going to be reserved for severe cases. And so what we can see is that we're trying to block in the first one a particular molecule that's found only in funguses, so that's ergosterol. And so by blocking that, we can actually target fungal infections without actually causing toxicity to humans. But as you can see, the very first one, the polyenes, are not very good at it. They bind agrosol and cause a membrane to leak, but they're highly toxic to humans. Azoles inhibit the synthesis, cause a membrane to leak. They're okay. And ultimately, the last one there inhibits the enzyme that's responsible for producing it. Cell wall synthesis. Some eukaryotes do have cell walls, like plant cells have a cell wall. Fungal cells have a cell wall made of something called chitin, and so there's particular drugs out there that are going to try to block cell wall synthesis as well as cell division. But these are going to have select toxicity, but luckily this drug actually for some reason is uptake by the fungal cells much faster than the human cells, so it still has some role to play in clearance of a particular infection. Nucleic acid synthesis, again, pretty poor drug target because it's going to be targeting all the same machinery that we have, except for there is one here that's taken up by yeast cells, so this would be good for yeast cell infections. Now the last ones are the helminths. These are the worms, and because these are usually found in places that are considered third world countries, there's not a lot of interest in research in terms of finding a drug that's going to work, mainly because there's no money there. There's no the medication is going to be unaffordable. So what incentive does a company like Eli Lilly or Pfizer or any of the other major drug companies, what incentive do they have to spend billions of dollars on researching a new drug if nobody's going to be able to afford to take it? So these are left pretty much to just treating as they come, not a lot there. So that's the end of chapter 20. So again, don't focus on the drug names as much. Please focus on the ones that are highlighted, but you don't have to memorize them all. You want to focus on the drug classes and how toxicity changes with each drug class. So if you have any questions, please let me know. Otherwise, I hope you enjoyed these two recorded lectures and also gave me the opportunity to try out my new toys for recording. So I will see you on Friday for the exam, and like I said, if you have any questions, send me an email.